In this video, we're going to discuss phagocytosis and the role it plays in the immunological response. So phagocytosis refers to the ability of a cell to engulf or eat solid particles, materials, even cellular pieces. There's several cells in the body which serve as phagocytes. That's going to be neutrophils, macrophages, and eosinophils. Neutrophils and macrophages are the most common. However, in parasitic infections, you'll see the eosinophils. Now, when we have the phagocytic response, one of the things you have to recognize is first that the antigen, represented here as this blue circle, has already made its way across our first line of defense. And let's just recall, first line of defense, it's going to be your cells, it's going to be the sweat glands with the salt, it's going to be the acids in the stomach, etc. So it has to get across that first line of defense into the body, and then the phagocytosis can begin. So when an antigen invades that first line of defense and enters the body, those invaded tissues are going to call immune cells to the site of infection through chemotaxis. The phagocytic cells are going to be the first ones to arrive, and then they're going to encounter that antigen, bind it in order to engulf it through endocytosis. So let's talk a little bit about that process of recognition from the phagocyte to that antigen. So when the phagocyte encounters an antigen, it's going to bind that antigen by recognition of certain surface markers or protein patterns on that antigen surface, and we call those pathogen-associated molecular patterns, or PAMs. And then those sites are going to be recognized by the macrophages receptors, and those receptors are referred to as PRRs, or pattern recognition receptors. And then they are able to bind the macrophage is able to bind the antigen and then begin that process of endocytosis. So let's look at those steps just a little bit. So here we have our macrophage, and here you're representing the antigen. It's going to be bound through the PAM and the PRR recognition, and then it's going to be brought inside the macrophage through that process known as endocytosis. And when it brings that antigen inside, it's going to actually form what we call a phagosome, so that it's going to be a membrane-bound vesicle containing that antigen. Now, also within the macrophage are something known as lysosomes, and lysosomes are going to have those enzyme and digestive enzymes within it. So what you will find is the lysosome will then fuse with the phagosome to form the phagolysosome, and then that antigen is now going to be exposed to all those digestive enzymes and antimicrobial agents within that lysosome. The enzymes, reactive oxygen particles, etc., or they're going to then break down and degrade that antigen, kill it depending on what it is, and break it down into pieces, as you can see here. Then the phagocyte is going to digest much of that antigen, but any of the remaining pieces, it's then going to spit out through exocytosis, and then the body can clear it. So that is the process of phagocytosis.